Thank you very much. It's a pleasure to be here. We will talk about antibody engineering, but from a different side today in many places is that we are developing mouse models perhaps for better targets for the antibodies that others like you are engineering and developing. Let me first give you a little bit of information about GemPharmatech. I won't read the disclaimer, I promise, is that we are really have the world's largest collection of genetically engineered mouse models, all created from one organization. And we have a major effort in R&D for new genetically engineered mouse models. The company started in Nanjing, China with our founder, Professor Gao Zhang, and it currently, within 4 years, now has over 800 employees. 5,000 new genetically engineered mouse models are created every year. Yeah, that number is true, it's 5,000. I've known this organization for almost 20 years, and it's really been remarkable what they've been able to develop. When I started some companies called Deltagen or whatever, we could do 250 a year, and we were pretty proud of that. But now that number has been blown away. And we now have started US operations and are also expanding into Europe to try to bring many of the products that we've created in China throughout the world. So we have over 16,000 gems that have been created. Most of these are conditional ready, where their LOX-P site has been floxed around critical exons, and then you can breed that with a Cree mouse and make your knockout of choice in whichever tissue. But primarily what I'll talk about today are just a few of the hundreds of humanized models that we've made to make, again, better models for therapeutic development. And what I'll focus on is the humanized CD3E mouse. So again, this is a slide taken showing Blincyto, which is really one of the first approved drugs using bispecific antibodies for the treatment of acute lymphocytic leukemia. You can see that the target of this, of course, is CD19. And as you see on the slide here, that this occurs very early in B-cell development, which is critical to try to catch what's termed, cancer stem cells early in that pathway. And then the other part of this, of course, is the antibody to the CD3 signaling molecule on T cells to activate the T cells and then generate a response to them. And again, I said this has been approved. So now what we'd like to do is try to look at, how can we make better models for both antibodies as well as bispecific antibodies. And humanization is the path forward, and two different ways to do that. You can take a mouse, transplant it with human peripheral blood mononucleocytes or human stem cell precursors, and then you can transplant the tumor, and now you can study this, and you can use your bispecific antibody within that mouse. Another way to do that is genetically humanize the mouse, and in this case where you put in a copy of the human CD3E chain, and then now you can just directly treat the mouse without having to go through the stem cell transplantation. And just to remind everyone here, here's a little diagram of the T-cell receptor complex with the CD3E. Again, you have the epsilon, delta, gamma chains that are here, and they're involved in signaling and stimulation of the T cells. And you can see this stalk region as highly conserved residues as well as a transmembrane region. But when you look at the human and the mouse, there's really a lot of disparity in the extracellular domains. And so the intracellular domain is highly conserved, but only 47% homology in the extracellular domain. So obviously using an antibody that's specific for human, the CD3E or other components is not going to work as well. And so the idea clearly is to create a transgenic model that expresses the human component of this. And this is just showing, as a background slide, that the mouse CD3E is indispensable for mouse T-cell development. This is one of the first molecules that was actually knocked out in mice. And if you do the knockout, you'll see that you nearly completely abrogate T-cell development in these. Again, as another sort of proof of concept here is that the CD3E, of course, is going to be indispensable for T-cell function. If you take a look at these same knockout mice and do tumor transplantation, you can see that now the tumors just grow, completely at will, as it would be. So again, very important for T-cell function, obviously. And so now what we've done is the first generation in our hands to humanize the CD3E. And we're doing this, as I said, with a back transgenic, and we're doing the epsilon component of the CD3 complex. And so what we chose was the bsAb background, primarily because there are a number of tumor models that are syngeneic to bsAb that you can transplant. And as you'll see later, we've also humanized targets within the tumors themselves. So now you can have the human target on the mouse tumor cells. And so what we did is, again, made this intact human back gene as a transgenic. And so now you see some of the validation data. 96% of the CD3E T cells express the human CD3E and the mouse CD3E. So one reason we did this is that we didn't want to disrupt the mouse immune system direct function. We wanted to express both the human and the mouse. And what you'll be able to see here is that quite clearly we have very strong expression of the human CD3E within these mice. And again, that's not present within the wild type. Again, the humanized model, just again to validate this, looking at the blood T cells, splenic T cells, you can see across multiple components, or looking at T cell receptor as well, where we get co-expression of the CD3E with the T cell receptor across many of the different components, again, both looking at blood as well as the splenic T cell counts. And again, the percentage of T cells, again, are between the CD4 and CD8 are normal. And then one of the last slides on this, just showing T cell activation, that now you can activate the T cells either with the anti-human CD3E or the mouse CD3E. And now you can see this activation component for either one, whereas within the control of the bsAb, it only works, of course, as you would expect, with the murine anti-CD3E. So again, I think it's a very robust proof of concept that these mice are expressing a fully functional copy of the human CD3E. And just going forward, looking at some other cell populations, the T cell, B cell, and NK cell populations are normal in these mice. And now you can see this, here's the CD4, CD8, as well as the NK cells in the human CD3, virtually indistinguishable from the wild type. And again, this was one of the reasons we wanted to start with the back transgenic. We didn't want to disrupt the normal function of the CD3E, but you'll see we have next generation molecules where we've done different replacements. And so now getting at some of the functionality, we can look at IL-2 and gamma interferon and TNF-alpha levels that are going to be released by these treatments, which is one of the hallmarks of CD3E. Any of you old enough to remember OKT3 was one of the first antibodies being developed, which caused massive cytokine release, which probably wasn't the best idea, but no one really knew then. But now you can see the release of all these cytokines in response, whether it's with the mouse or the human, and they work pretty equivalently. And so now getting to the real crux of the matter is, how does this work with bivalent antibodies? This work was done in collaboration with Crown Bio. We have had a long relationship with them. A long relationship, it's a 4-year-old company, but it seems long, right? There we go. So now you can take a look at a PBS control, and now you can treat these mice with either the murine or then the fully chimeric molecule. And now you can see that when you do this with bsAb, and if you're looking at the CD19 plus B cell count, you can see that in the bsAb this is not affected, but now you can basically deplete the B cells within these, so showing the bivalent antibody is targeting the normal B cells because it's going against the murine CD20. And now when we look at the A20 growth and the growth inhibition, you can now start to see here's the control with PBS, and now as you start treating with the bispecific antibodies, you see a marked reduction in the tumor burden over time. So what other things are we looking at? One of this is going to be the next generation. As I talked about, we did the CD3 epsilon, but the CD3 also has other components which are present on different subtypes within T cells, and actually even on NK cells or gamma delta T cells. And so this could be different. And again, we want to make research tools available to people who may have different antibodies or different targets or components of the CD3 that they'd like to stimulate or use for tumor attack. So this now is again, this is another large transgenic expressing all of these components, and now you can see the Western blot where you can see the epsilon, delta, and gamma are expressed in the mice. And then if we look at the co-expression of these proteins in these heterozygous mice, again, you can see that, within the heterozygous, we see the expression on these cells, and again, absent in the wild-type mice. And then if we take a look at therapeutic CD3 antibodies, I won't mention them, I don't think, by name on this slide, but now we can see that combined to mononuclear cells in the peripheral blood of these mice. And again, as we saw on the previous slide, now with this more complex transgene, again, we see similar B cell, T cell, and NK cell activity here. In the heterozygous mice, NK cells are still present, no different, and again, we're seeing basically normal immune competent as wild-type mice. And again, if we look at just in vitro cytotoxicity, again, just titrating the antibody, we can now see again that it's working in vitro. And of course, now when we go into the tumor model with these bsAb mice, which have been transplanted, you can now see virtually wiping out the tumor in this case. I won't go to any detail on this. I don't want to do too much of an advertisement, but one of the things that we did, which is a bit unique, is that we did this on the bsAb background. And one reason for doing this on the bsAb background is that you now have a host of tumor models that have been well validated for more than 30 years that can be used in the bsAb background. And we've also humanized different components which are targets for potentially immuno-oncology within the tumors. So you can now get the appropriate tumor to transplant with your bispecific antibody. And so where are we now? Again, this was our first generation. We have the second generation, and now we're looking to make homozygous mice for these to make everything easier for the whole breeding scheme. So I know that was a little rapid, but I thought a lot of that is background information. We'd be happy to share any more information with you. We also are interested in other targets in bispecific antibodies. We may have that more of a discussion at the roundtable later today. But if anyone has ideas or that they would like, we also do custom mouse generation. We work with researchers around the world to make the models that they need. Another area, again, obviously, where we are with the COVID outbreak and the pandemic, very early on, we started creating mouse models for studying the SARS-CoV-2 research and created humanized ACE2 mice. As I'm sure most of you are aware, mice do not become infected with this virus because of the difference in the ACE2 receptor, which is the binding for the spike protein. So what we were able to do is create transgenic mice. We did sort of the K18 promoter on the human, but we also created full-length knock-ins as well as chimeras, where we basically just changed the extracellular domain of the ACE2, leaving the cytoplasmic domain intact. And we did this on BLAC6 and bsAb to, again, allow researchers to have the resources that they need. And again, if we take a look at this, I'm sure I can't see it, so I doubt if you can see it. But this is just looking at the mRNA levels and the characterization of this K18 transgene. And now you can see in the tissues that are reflective of basically human infections as well, looking at the lung as well as the pulmonary airways, you can start to see expression of the messenger RNA there. And we also can go back and look at the viral RNA within the lung. And again, this is now the full-length knock-in of the human ACE2. Again, we're now able to see the appropriate expression of the virus particles upon infection and also see the virus burden within these, as well as the production of potentially neutralizing antibodies. So we actually were very happy. This was a paper that came out, I believe, in February, published in Science. And so what this was able to do is take the mouse model and use small molecule inhibitors for viral infection and activity, looking at this transgenic model. We didn't get to be a co-author, but if you dig deep into the materials and methods, you can see that it's our mouse. So sometimes you got to dig a little deeper. Because first thing you do is you look at the paper and go, oh no, I got a competitor, but then you realize, oh no, it's us. That's good. That's the good news. But now you can start to see now as you look at the infection, again, from their work, looking at their different components and compounds, starting to see the inhibition. And again, this is really only possible by having the humanized version of this. As I mentioned, we were making different knock-in models and making full-length full-length ACE2, but we also made a chimeric molecule where we just changed the extracellular domain. And again, now this is a little larger, so maybe you can see it, looking at the different tissues where you see infection, and again, reflective of what you see in the human population, and very similar to our full-length human, but now you have the cytoplasmic domain of the mouse molecule to ensure proper functioning of the molecule. And then, as I mentioned, different flavors for different experiments. We've also done this, as I said, both on the Black 6 and the bsAb mouse background, and again, just showing the same kind of experiment that you've seen before, that this is working well. One of the advantages of having a large research and development group is that we really can make transgenics, knockouts, knock-ins on virtually any mouse strain. And that's really important. If you have complex transgenics, complex humanization, you don't want to start over with a 3-year breeding program. It's nice to start out with something that already has the molecules you're interested in. Another part of this component is looking at the TMPRSS2 molecule. This is the endogenous protease that's involved in cleaving the spike protein to ensure viral entry. Currently, there's just the mouse version, but as people are developing small molecule inhibitors for this, we've actually moved ahead and knocked into the endogenous locus the human cDNA for the TMPRSS2, and these mice are now becoming available, and of course, making them with our ACE2 is a fairly obvious thing to do moving forward. And something which everyone may not be aware of, but there's also candidate second receptors for the SARS-CoV-2 that can promote infection, particularly in pulmonary and bronchial epithelial cells. And this is a molecule called AXL. And this, we've already moved ahead and now made a chimeric coding region for this and created the mice and knocked this in. So I may have gone through that quickly. I tend to talk quickly, but I just want to say that, one of our goals is to, wherever there are going to be human disease issues, where there are mouse models that are needed, we'll proactively move ahead and make the models, assuming and hoping that researchers will come forward and using them, because there's no time to wait. And we've already made, as I say, a number of these for the COVID area. And so now I just want to say is we look forward to collaborating with researchers from academia and companies. We have a lot of mouse models. We have the capacity to make a lot more. And we want to be able to do that to really advance therapeutic research. So thank you very much.
Okay, so we'll just wait for the Slido app to load up for any questions. So those of you joining us digitally and in person, we use Slido, as you're probably aware, for all of our questions. So if you do have any, if you could submit them through now. We have around 10 minutes to go before the end of this talk, so we have enough time for all your questions if you do have any. So Mark, I'm going to start with the first question. The first question is from Dale. Thank you for the talk. What are the average timelines for custom knock-in mouse generation?
Well, for the custom generation, it's between 3 and 6 months, and there's that window of an extra 3 months because that's the mouse breeding cycle. It depends on when you would like to get the mouse, what form you would like, a simple transgene, but usually 3 to 6 months. We can make a design in probably 24 to 48 hours for you and start the project right away. And if we have something that's already frozen, our entire collection is frozen either as sperm or as embryos, and those can be reanimated within a couple of weeks, and those shipments could potentially go out in 8 weeks once the mice are old enough.
Fantastic. Moving on to the next question. T-cell bsAbs are surely a hot area. We have heard at least 2 talks on T-cell engagers / recruiters beyond TCR, such as 4-1BB and PD-1. Have you thought about bsAbs?
Yes, a bit. As the intro said, I'm a recovering immunologist, so we give this some thought. Because as you look at bispecific antibodies, there are two edges of this sword. You can use it to target tumors, and you may also want to have different ones that can damp down your immune system for autoimmunity. So the complexity of molecules has clearly grown since I got my PhD, and I think the ability to really specifically go into T-cell subsets, NK cell subsets, and really fine-tune your response is really just starting. So I think there's a plethora of potential target molecules that we could use, depending upon what you want to target and the disease you're treating.
Fantastic, thank you. There's still 6 minutes to go, so if you do have any additional questions, please put them through. How can mouse models such as the ones you described help study the long COVID symptoms, which seems to be quite subject-specific and less common?
That's a great question. Throughout my career, I've always tried to do long-term studies in mice. I think that's really important because basically all of our mouse research is done on juvenile mice. And in the human population, that's not a population that gets very sick. But every time I would go to the National Institute of Aging for the NIH, they didn't want to fund the long-term studies. I go, well, where else are you going to go but the Institute of Aging? So I think it's really important to do this, but I think some companies may have to take it on their own nickel. We would see, but I think this is going to be critical, and that's one of the reasons we created this suite of the ACE2, but also the TMPRESS2, so that the virus is going to be properly processed. We also need to make sure that I think that we adequately keep the mice around long enough to be able to do those studies, but I think that's critical for long haul.
Fantastic, thank you. You've been a great group with Slido, so thank you. Could you cross your humanized mice with a disease model, mouse model, specific disease therapeutic area testing? So I'll repeat that because I messed up so much.
Yes, no, absolutely. We could do the crossing, but as I said, it depends on how complicated it is, because one thing I didn't mention is that, we've basically made humanized knock-ins for all immuno-oncology targets. I'm not exaggerating, there are 200 or 300 that we've done. And so we have double, triple humanized models, PD-1, PD-L1, CTLA-4. And so if you have the triple combination, it's about a 2 or 3 year breeding program. So what we might do if you have a different disease therapeutic model, might just decide to make that transgene model again on top of our existing models and save 2 or 3 years of breeding time. So a little bit of a technical breeding question, but it's important. But yes, we could either cross them or we could just make it over, and often in the old days people would make a transgenic model and that's what you're stuck with. Now, as you see, we can do 5,000 a year, so we can put it on the better mouse genetic background that the researchers may need.
Fantastic, and I don't see anyone typing on the phone, so I assume there are no further questions. We are a little bit early, 5 minutes, so does anyone have any final questions at all that they want to submit through the app? Going once, going twice, gone.
A speaker never gets in trouble for ending quickly, right? It's going over.
Well, thank you, Mark, for joining us today and giving a presentation on GemPharmatech and your solutions. They are at the booth just outside on the right, so if you do want to continue your conversation with the company, you can speak with Mark and the team. They're just behind you as well.
Well, thank you very much,
Mark.
and thanks for your attention.